Hi, everybody. My name's Eric. I'm an MD PhD candidate in the Farber Lab at Columbia. I am happy to present a little bit of my data from my project titled Tissue-Directed Maintenance of Human Memory B cells in Vaccination and Infection. About a year ago is when I tuned into the Immcantation seminar for the first time. That was also around the same time that I got my first bit of AIRR data, and so it's been great to learn from other people. And then I'm also very happy to be able to kind of contribute back to the community and get feedback from people. So when we think about vaccine responses, at the core of pretty much most vaccine responses and infection responses is the activation of naive B cells to enter germinal center reactions and go through a maturation process to differentiate into antibody secreting cells and memory B cells. But this process really in the primary response is quite minimal, and it's not until a secondary challenge, either through a booster vaccination or through an actual infection, that those expanded memory B cells are re-engaged and expanded into long-lived plasma cells and memory B cells that provide lifelong immunity. And so these cells are really, really important, particularly long-lived plasma cells, because they're the ones that directly maintain those circulating antibody titers. But for the purposes of future booster vaccinations and permitting the immune system to adapt to new challenges, it's the persistence and quality maintenance of these memory B cells is really, really important in the body. A major challenge to understanding how exactly these memory B cells are maintained, particularly in humans, is that the vast majority of these cells are maintained not in blood, but actually in tissues across the body. And as is the case for other lymphocytes like T cells, B cells can establish long-term residency in these sites, and they express unique markers and chemokine receptors that hold them in those sites. And in the last about 5 or 6 years, it's been well established that in mice that B cells in tissues can perform site-specific functions, optimize responses to local antigen, and secrete antibody more quickly and qualitatively in different ways than those in circulation. But despite that research, our understanding of the features of the memory B cells and their maintenance at homeostasis across human tissues and across human life is still relatively poorly defined. And so we wanted to ask the question, how exactly are human memory B cells maintained and organized across the body? To tackle this, we have the benefit of maintaining a relatively large cryopreserved human tissue resource in collaboration with local organ donor services. And so we use this resource to embark on a combined flow cytometry and single cell sequencing project to essentially examine the diversity of memory B cell subsets and clones across the body. And so I'm showing you here just some data from that. To start us off, this is essentially 104 donors profiled by flow cytometry, where we have a variety of different lymphoid tissue sites and lungs, so blood, bone marrow, spleen, and lung-associated lymph node and mesenteric-associated lymph node. And what you can see is that really the blood has a very small percentage of CD19 positive B cells. And amongst those B cells, a minority are actually memory. Instead, the vast majority of memory, like I said, is found in lymphoid sites like the spleen, the lymph node, and the lymph nodes. Now, memory B cells come in a variety of different flavors, and I just wanted to highlight a couple here. So the kind of canonical germinal center product are these classical memory B cells that have these classical memory markers on their surface and that are highly responsive to T-dependent signals like IL-21 and IL-10, re-enter germinal centers, differentiated plasma cells. They can come as class-switched or non-class switched versions. But in the last few years, there's been a particular amount of attention paid to an alternative subset of memory cells called atypical memory B cells. They're called that because they, instead of expressing those canonical memory markers, they express inhibitory co-receptors and integrin markers like the CD11C and FcRL5 receptors. And instead of those classical T-dependent signals, they respond to highly inflammatory signals like interferon and toll-like receptor agonists. And these guys have really been described mostly at homeostasis in patients with autoimmunity, in chronic infection models like HIV and malaria, in certain populations in old age, and recently they've been described at higher frequencies in SARS-CoV-2 responses as well. And so we wanted to just kind of get an overall view of how much does the composition of these memory B cell subsets differ across different tissue sites. And so I'm showing you here flow cytometry data where we've identified 4 different broad B cell subsets, naive B cells, class switched classical memory B cells that are switched and CD27 positive, non-switched classical memory cells, and then this weird atypical memory subset. And what you can see is that there is quite a bit of variation from site to site. And so really the lymph nodes, this blue and green, and the spleen are the primary sites for that classical memory. But the spleen also is unique in that it has an enriched pool of these non-switched memory cells. And the bone marrow really uniquely seems to be a reservoir for these strange atypical memory cells. And so importantly, we wanted to also understand how exactly vaccine and infection-induced memory really varies across these sites as well, because that's really the ultimate question that we're trying to tackle here. And so we used vaccine antigen tetramers against BCRs in flow cytometry that are fluorophore conjugated to identify a variety of different common vaccine and infection-induced associated antigens. I'm showing you here some of the phenotyping within each of those antigen-specific pools. And really the takeaway here is that there's quite a bit of diversity across tissues, but also across specificities within these antigen-specific pools as well. So, for example, in this first plot up here, I'm showing you the the isotype composition of these different memory B cells. And you see that these infection-associated memory pools are highly, highly class-switched. They contain very, very few of these non-switched memory cells. But then these childhood vaccine antigen-associated pools like measles and tetanus contain a much more diverse mix of class-switch and non-class-switch memory. And tetanus in particular, regardless of the tissue site, seems to have just a remarkable amount of non-switch memory maintained across life in these adults. We also looked at these atypical memory cells, and we found that, again, all sites or all specificities tend to be enriched for them in the bone marrow, but really, again, these infection-associated pools are highly, highly enriched for these atypical cells, suggesting that there's really some kind of alternate functional specialization in these responses compared to other traditional vaccine responses. And so having identified this diversity across the body, we wanted to next ask the question really, we wanted to really tackle this question of how exactly vaccine memories maintain in our bodies in 2 more specific parts. And this is what I wanted to share with regards to its relevance to incantation. So the first is whether or not antigen-specific memory B cells from various lymphoid sites vary in their transcriptional features and subsets. Can we identify the same amount of diversity at the transcriptional level? And then, is that diversity interesting in how it's clonally related across tissue sites and subsets? And so to achieve this, we performed paired single-cell sequencing of memory B cells sorted from the bone marrow, spleen, and lymph node of 3 donors in our cohort. And specifically, we sorted both polyclonal memory B cells and those antigen-specific memory cells using hashtag conjugated fluorescent multimers. And so the way this works is that all of them are essentially put on the same 2 colors. We can sort those out by FACS as our antigen-specific cells. And then those oligo hashtags allow us to deconvolute the exact specificities of those cells in silico afterwards. And so, for example, this is just a representation of what that deconvolution looks like, for example, for the tetanus cells based on their unique hashtags. And so from this protocol, we essentially were able to isolate about 44,000 memory B cells, 6,000 of which were antigen specific for one of those four specificities. These are, again, distributed across donors and those 3 tissue sites where you can start to see some tissue-specific patterns. And using a combination of unsupervised clustering and supervised classification, we identified through this process 3 major transcriptionally distinct memory B cell subsets. And I've termed them here classical memory cells, this interesting marginal zone-like memory cell population that I'm going to talk about a little bit more, and those atypical cells that we saw that were highly enriched in those infection-associated memory pools as well. Their transcriptomes were highly distinct, so by differential gene expression, you can see that classical memory cells express CCR7, which localizes them to secondary lymphoid sites, as well as the stem-like factor TCF7. While atypical cells have that kind of strange ITGAX, which is CD11C, and those FCRL receptors expressed at baseline. And so lastly, we also have this marginal zone-like population, which was a little bit of a surprise to see. These cells are a splenic population that have been described much more extensively in mice, but they express a notch-driven signature and express large amounts of complement receptors on their surface and are typically associated with non-protein antigens and other non-traditional antigens. And so you can see some examples of the UMAP annotation here. To validate some these these classifications, we used GSEA, and we compared these differential gene expression signatures against published signatures of marginal zone B cells from the spleen from this paper, as well as memory B cells in circulation that are specific that were induced by HIV and chronic malaria infection. We found that the signatures were largely consistent with those subsets identified in other papers as well. So that was nice to just see that we're looking at robust subsets. So we examined what exactly the tissue distribution of these different subsets were. And what we found is that each subset was actually remarkably compartmentalized within each of the different tissue sites. So for example, those marginal zone cells, which I mentioned have been previously described in the spleen, were highly enriched in the spleen in all three of our donors as well. The atypical cells were consistent with the flow cytometry, enriched in the bone marrow. And the classical memory cells, while distributed across the body, were really predominant within the lymph nodes. When we looked at this within our antigen-specific pools, we saw that those patterns were largely preserved, so those same tissue patterns, but that individual antigen specificities still also deviated with their own kind of unique patterns. So really probably the most prominent thing here is that SARS and flu, just like in the flow cytometry, really are highly enriched in the bone marrow, but also other sites for this atypical subset. And so while the prevalence of classical and atypical memory cells in vaccine responses was relatively expected, here we were relatively surprised to see that this marginal zone subset was so abundant within the vaccine responses. This is just a little bit of context. In mice, these cells are typically defined as innate-like B cells that develop from distinct precursors than classical cells, and they typically are associated with distinct antigens, like non-protein vaccine antigens. And consistent with this, their BCRs are usually not mutated. But in humans, those developmental origins are a little bit less clear. And so while they're still typically reported as a non-switch memory subset, they've really rarely been implicated in vaccine responses. And so just to kind of confirm that what we were seeing is real, we validated this by flow cytometry. And we saw that looking at these two markers identified by our DE, CD300A and CD1C, the spleen is really the site, again, that has the majority of these cells. And within that splenic population, there's quite a bit of variation from antigen to antigen in the prevalence of these different marginal zone subsets, but they're all present at relatively respectable frequencies. Okay, and so now that we've kind of defined these three major distinct subsets that are kind of tissue confined, a major question that we had was, how exactly are these cells related? Canonically, we might say that the classical cells are really like the T-dependent germinal center reaction product, whereas these marginal zone cells and atypical cells are historically associated with extrafollicular T-independent responses. But more research recently has kind of called this into question, and now it's not really clear whether these are really distinct precursors and lineages driving these identities, or if it's actually diversity within the same clonal lineage. And so to interrogate this, we looked at the repertoire analysis for the cells. We annotated essentially about 3,000 clones within the antigen-specific compartment. And we found that these clones were really highly expanded relative to the rest of the population. And when we looked across these different specificities, we also saw that there was quite a bit of variation in the degree of clonal expansion. So those childhood vaccine antigens that have been around for a long time were really highly expanded, with tetanus in this donor being up to like 80% of its clones participating in expanded clones. But regardless of the degree of clonal expansion, we noticed that for all the specificities, these expanded clones, represented by the numbers here, were really highly disseminated across the body. And so you can see that, so here the overlap between the circles indicates presence of that clone across either tissue site. So 21 clones in the measles population were present in all three of our tissue sites. And so we asked the question, how exactly does this tissue distribution then associate with the identity of these memory subsets? And we found something quite interesting. So I'm showing you these kind of complicated, what we call clonal distribution plots. And essentially what you're seeing here, I'll break it down, is each specificity displayed on a grouping of circles. A single column here is representative of one clone, and the pie charts represent the phenotypic distribution of those clones across the three tissue sites. And what we observed really remarkably is that even within a single clone, we had representation at high frequencies of all three of those distinct subsets. But even more than that, those subset identities were still compartmentalized by the tissue. And so the bone marrow still had the atypical cells, the spleen had the marginal zone cells, and the lymph node had those classical memory cells. And we saw this was kind of reproducible across all the largest clones across all 4 specificities, which was really remarkable. When we did this with isotype, we saw a different pattern. So by isotype here, the coloring represents either IgG, IgA, or non-class switch isotypes. We saw that clones tended to be either all of one isotype or another, and that those didn't necessarily always correlate with the identity of the memory B cell subset in that same clone. And so, for example, probably the most remarkable example of this is that in tetanus, all of the largest highly expanded clones were entirely non-switched. And so in the tetanus memory response in our population, there must be these really highly expanded non-switch memory clones that are just kind of sticking around for the long term. Okay. And so lastly, we did a little bit of quantification of the mutational load of these different memory subsets and across our different specificities. We found that as expected, the isotypes varied in the degree of the average degree of mutation of their BCRs. Notably, we found that although in all these specificities, the non-switch memory was the least mutated. In SARS, the degree of mutation in the class-switched memory cells, the IgG memory cells, was roughly on par with the degree of mutation in the non-switch memory cells for tetanus, indicating there's quite a bit of deviation here from what the expected norms are for these different canonical subsets. We also did the same thing by subset, and we saw similar patterns. Notably, although there is some differences between marginal zone and classical memory cells, all 3 of these subsets had relatively mutated BCRs, suggesting that they've all participated in some degree of affinity maturation. And so the last question kind of that we wanted to ask here is if we could clarify the relationship between these subsets within the clonal lineages themselves. And so far I've shown you that within an antigen-specific response, you can have a diversity of memory B-cell identities that are maintained long-term in tissues, and that within a single clone, those same diverse subsets are still maintained. But we wanted to know whether within that single lineage, is subset identity restricted asymmetrically to certain branches? And so you can imagine on a phylogenetic tree here that you could have a few different hypothetical patterns for how those subsets are distributed. So this is a measles clone. I've kind of taken away the coloring for a second. But one possibility we hypothesize is that you could have a situation where subset is confined within certain branches, indicating that subset is an irreversible change within a lineage. Alternatively, you could have a situation where the subset is associated with a degree of somatic hypermutation or the duration of germinal center participation, therefore still ending up with diversity within a clone, but asymmetric distribution of those subsets. What we found in practice is that in all of our antigen pools, this kind of asymmetric distribution was not present. Instead, those subset identities were highly, highly distributed across the entire tree. And so I'm showing you 3 trees, one from each of the largest clones in the 3 specificities. And you can see that there's no real association with the position within the tree and the subset. Instead, if you were to sit here and kind of take a stare at these dots, you'd see that the association is again more between the tissue location and the subset, indicated by the shape and the coloring. And so just to summarize a bit from what I've shown you here, we found that lymphoid tissues maintain unique pools of transcriptionally unique vaccine memory, likely with distinct functional requirements and responses. For a given clone, the memory B cell subset identity can be heterogeneous and a feature of tissue, and that antigen appears to in part drive the isotype of the clone. And then importantly, cells from all 3 subsets, despite what their canonical definitions say, demonstrate features of germinal center experience, including the presence of long-lived expanded clones and mutated BCRs shared within a single clonal lineage. Okay, and so altogether, these data demonstrate that there are likely underappreciated reservoirs of vaccine-specific memory in these various tissues that probably have distinct requirements and responses that should be considered for future efforts. All right, so just a few acknowledgements to my lab and then also some of our collaborators and to the donors and their families for their contributions.